And I will be posting this by tomorrow, hopefully by tomorrow morning on Zoom. And I will put the links in the Slack room also so that you can access them a little bit more easily. I'm really happy this morning to be introducing Andrew Givone and Miko Mallari, who are were participants in the January hackathon. And I know both of them have used antibodies in their internships and in some cases work experiences. And so I'm really happy to learn about that more. So what we're going to do is I will have each one of them talk for a few minutes and then we'll have questions, open it up to questions. We're also recording this for Talking Technicians, which is a really cool ATE podcast that has stories of all kinds of people working in technical careers in different kinds of jobs. And I really highly recommend it. It's really good. Okay, so Andrew, how about you go first?
All right. Thanks for the introduction, Sandra. So yeah, I'm Andrew. I attended the hackathon in January. I was on Dr. Brian's team, the Breaking an Antibody team. So that was really my first exposure to antibody engineering. We were using IC3D to view the antibody-antigen interaction and using that program's mutation functions to visualize which amino acid residues we could mutate to potentially break that antigen-antibody interaction. So after that, I got really interested in antibody engineering, and I attended the Wistar Institute's biomedical technician training internship. It was a 12-week course that just finished last week. And yeah, so through that, I got to intern at two separate labs. The first one was a company, Intermolecular. They do all sorts of work on antibodies. They have discovery teams, characterization, production. The team I interned on was the shotgun mutagenesis epitope mapping team. So using shotgun mutagenesis and alanine scanning to identify the epitope on an antigen. A lot of it was SARS-CoV-2 because of the pandemic. And then my second internship was in a lab at the Wistar Institute, the CULP laboratory. And there I used another protein modeling viewing software, PyMol. And in that one, I was modeling an influenza B protein that could be used for immunization, for trying to find antibodies for potential therapy for the flu. And so after that, I actually just received a full-time job offer at Intermolecular, the first company I interned at, which is very exciting. And there I'll be joining the MPA team, the membrane proteome array team, which uses a matrix of cellular membrane proteins, so about 6,000 cellular membrane proteins. And they use that to scan antibodies and try to identify any off-target binding for those antibodies. And yeah, I'll be starting that in about 2 weeks. And that's pretty much all my experience with antibodies, my background, my work, and what I'm going to be working on. And Dr. Porter sent us a list of talking points here. One of them was, now that you're graduated and working, what would I have liked to know before I graduated? And I think the biggest thing for me is I would have liked to know that I don't have to know everything. In school, it's a lot of memorizing and like preparing for tests and like knowing all the tiny details about certain things. In actual lab, you like do have to know those specifics, I feel, but also you can use a computer to look up anything you don't know whenever you want. There's tons of people there. You can ask questions if you don't know anything. I think it would have been good for me to know that the environment is like much more collaborative and that I don't have to know every little thing by myself. Yeah. Does anybody else have any other questions?
I think, Andrew, we'll hold the questions until after Miko talks, and then we'll come back to those. Thanks.
Yeah, no problem.
Miko, why don't you tell us about your experiences?
Sure. I actually made a little slide deck. Is it cool if I share that?
Oh, of course.
Pull it up. But anyways, yeah, my name's, you can see my screen. My name's Miko, as Sandra pointed out, and I'm located in the San Francisco Bay Area. This is my second hackathon, and I'm really excited to continue working on the project that I worked on in January, the IEDB team. And currently, both a research associate at Sana Biotechnology and also a Catalyst intern at UCSF. I started at Sana just over 2 months ago, but due to legal reasons, I can't really talk too much about what I do at either of these jobs. So I kind of made this talk more focused on how I got to where I am today and how I used antibodies in some of these other jobs. Feel free to do a quick web search if you're curious about what these places do. I'm not really supposed to talk about it. And I know that there's a wide range of experience of participants in this hackathon, so I hope some piece of my story kind of resonates with you guys. So I'm going to walk you through my work history and talk about how all my experiences landed me where I am today. I grew up in California, specifically here in the Bay Area, and I only left to go to school in Southern California to pursue my interests in environmental chemistry and studio art. And I know that there's a lot of early career people in this group, so I thought I'd include some fun things to do, maybe to connect with you guys, make it seem like researchers are real people too. So when I was in high school, I was a Boy Scout. And in the beginning, it was just something that my older brothers did. So I did what a good youngest child does and just kind of followed my brothers around. But I grew into it and I really liked it. I followed it all the way through and I actually qualified to be an Eagle Scout. I had a lot of fun and I learned a lot about the little things, and those experiences really fueled my love for the outdoors. I'm also a pretty active person. I've been diving since I was 8 years old, and I was fortunate enough to go to college where I could be a part of the swim and dive team through my 4 years there. But I find it hard now to find diving boards around, so I channel my energy in like other directions. I'm a big rock climber, and I really enjoy like the adventure element out of it. So it gets me outside of the lab sometimes, which is nice. As I mentioned, I also studied studio art in college, and I first fell in love with photography in high school and still carry a camera with any adventure I go on. And I guess this was like a really good opportunity to plug my photography. So bear with me. Here are a few examples of projects I did when I was developing as an artist. During my experimentation, I became pretty interested in how images can portray different stories and ideas. I really enjoyed the fact that lighting could really evoke emotion and tell different stories. I tend to like portraiture as a medium because I find out a lot about people when I work with them. And I firmly believe that my interests in the arts have allowed me to develop my skills to convey ideas in a more compelling manner. But okay, I'll get back into my career stuff, but here we go. So summers during college, I worked at a small clinical research lab, mostly doing assisting on the clinical side and administrative side. And following school, I decided to continue out helping there. For the longest time, I thought I wanted to be in healthcare. So I was trying to get some unique experiences while trying to take some postbac classes before I applied out to some more traditional healthcare related programs. I was at Provenience when I saw a huge potential in biotech, but I didn't really think I had the skills to be successful to get into a research position in the field. While I was taking some of those postbac classes at CCSF, I discovered that they had a biotech program, and I probably took the biggest risk of my life and changed my direction again. But I'm really, really happy that I did. I'll admit it was a little difficult at first because but there were so many awesome people I met in the program. My very, very first lab partner is actually in this hackathon too. So shout out to you, Carmen, for helping me out through some of those tough times. So I stuck through it, and I really felt I'd found a home in this program. And I finished a number of the program offerings there. And I cannot stress the amount of opportunities I found because of this program, including my involvement with this hackathon. My involvement will become pretty obvious pretty soon. While enrolled at CCSF, I got connected to my very first research lab experience at UCSF, studying topics in cancer immunotherapies and antibody engineering. I was fortunate enough to work in Dr. Jim Wells' lab with Dr. Jamie Burns and Dr. Susanna Elledge. Susanna was finishing up her PhD at the time, so I got to work with her a lot and got to see what life was like in an academic lab as a student. I also got to taste what like a really great lab culture could be. I'm not going to dig too much into this project, but in this project, I engineered protein products called bispecific T-cell engagers, or BiTEs. And these BiTEs would allow one to connect a T-cell to a cancerous cell and activate them for cancer killing, as you can see here. I think this QR code is still live if you want to hear more about this project. You can try clicking on that later. It was this project and this poster I shared at the ATE Principal Investigators Conference in the fall of last year. And at that conference, it was that conference that got me involved with this hackathon. Todd actually stopped by and asked me if I wanted to participate in a hackathon. And honestly, I did not really know what that meant, but here I am again for the second time. So thanks for the invite, Todd. Following that experience, I wasn't entirely sure what the next steps for my career should be, but it got me really excited to be a part of biomedical research, and I applied everywhere, jobs in industry, in academia. I even applied to grad school, and I was also offered an extension at my current job. So I got into grad school in Ohio, but I opted to defer that to participate in the California Institute for Regenerative Medicine's Bridges program that was connected to the biotechnology program at CCSF. And you might be wondering why I just didn't go to grad school. I decided that I wanted to really make sure that research was for me, and if I could really last in research. This program through CIRM was a funded research opportunity that put me in a position to find a lab that is studying topics in stem cell and regenerative medicine that I was also interested in. Well, it was tempting to just head off to go to grad school. They were not like offering to help me pay for school or even find a lab to work in until year 2. Through CIRM, I was able to conduct real research, real funded research at Vitalant Research Institute with Dr. Marcus Muke. There, I helped narrow down some of the mechanisms in the initial transplantation of hematopoietic stem cells into immunodeficient mice. We were trying to improve the establishment of a human immune system within a mouse. This would allow one to have more representative in vivo studies. And we used antibodies a lot in this project to identify cells, our cells, and if our cells were actually able to be transplanted. In this diagram, you can see we used flow cytometry to sample tissues and blood to see if our human cells were actually able to survive in these mice. These antibodies would bind to only human cells, and if we observed any that were CD45 positive, we deemed them as human cells. If they also expressed CD34, we called them like an early progenitor cell. And if they had all three of these markers, we believed that those were hematopoietic stem cells. And this is just another cool application one can use for antibodies. I wish I included a QR code for this poster too, but I forgot. But you can reach out to me at some other point if you want to hear more about this. I felt like I grew a lot in these internships, and that I was able to understand what it takes to do research, or at least I think I have a little taste for it. I was faced with a tough decision to continue with my lab or find one more experience before the next grad school application season. I ultimately decided to join an industry job because I had experience in an academic lab at UCSF, a nonprofit lab at Vitalant, and I thought that an experience in industry at a company whose work was a little closer to the clinic would provide me with a more well-rounded understanding of the biotech field before I actually committed to grad school. I didn't completely go into industry though. I still participate in things like this hackathon and the Catalyst program at UCSF. I definitely have to be careful working in industry. Because with things that I talk about though, so sorry I can't talk too much about my experience there, but yeah, it is what it is. Anyways, let's see where am I? Oh yeah, sorry. Next, I think I have another slide. Oh, okay. Yeah, okay. I skipped some stuff. Anyways, this experience into research so far has really been good for me. I've been in school for a while, and there's way more stuff, as Alex mentioned, that you can learn than in your textbooks. In research, you can really dig deep into subjects that really interest you. I know that it seems like I have a ton of experience, and maybe you're still in high school, maybe you're still an undergrad, or you're a postgrad just trying to figure it out like I was. I will say that all of my relevant experiences has come in the last few years after I completed my undergrad. And before I got involved with these programs, I was having a hard time finding opportunities that were not connected to programs like CCSF. So to all of the instructors that are at this conference, thank you for participating in things like this and bringing these ideas and connections back to your home programs. I owe so much to the City College of San Francisco's biotech program, and I definitely would not be where I am today without them. But tangent aside, I wanted to get to some other pieces of advice that you may be interested in if you're interested in research and you're still kind of early in your career. If you choose to get into research, it's important to develop good habits in order to be successful. There's a really big need to be self-motivated because there are a lot of things you need to do, need to learn, and you're probably not going to get it on the first try. You're going to fail. So always keep in mind like why you're doing this. A big reason I deferred grad school was to figure out if I was cut out for research life, and I've definitely learned a lot about myself and my work ethic in this time. Another big tip would be to get to know the people in your lab, invest some time into these relationships. They can really help you out. You never know when you're going to need advice or like an extra hand. Also, network all the time. It's a really good skill to learn, and you can find so many opportunities that way. Next, I'm a huge proponent for the work-life balance. It's a really tough job, and if it wasn't for my hobbies and the support I have, I don't really know where I'd be. Finally, my last tip would be to take your time. Find events like this to be a part of. Find some good mentors. See if there are internships you can apply to before really taking a deep dive into your career. You don't need a full-time job, if it's your first experience in research, you may find you don't really like it, or maybe that experience just puts a bad taste in your mouth. Internships are great because you are literally there just to learn. No one is expecting you to know everything at the beginning. I've made so many mistakes in my entrance into the research field, but I've realized it's all just part of the learning here. And I'm really glad I took my time and eased my way into it. Also, I guess one last thing, really leverage your time as a student. There are a ton of resources and programs out there that are just made for students. And a lot of those things can go away when you step away from a program. These experiences have really motivated me to, or have really motivated me and confirmed my interest in pursuing grad school, and I'll be applying out in the fall. I'm currently figuring out what other experiences may prepare me for the long haul that will be grad school and ultimately my career. And I'm really interested in studying protein and antibody engineering for therapeutics. I'm really fascinated by the versatility and specificity that engineered antibodies and protein products can have. I do think there are so many applications for these systems, and I hope to find a lab that I can continue to explore this. Even being part of the IEDB team in the hackathon has changed the way I've approached some problems. I have even made suggestions that I've justified using the IEDB to some projects that I've been a part of since I've learned about this tool. And that was kind of a cool moment for me to make that connection and think about how we can use some of these tools in my own research. So I'm really excited for where my career is heading, and I hope to continue learning with all of you. Happy to take questions, and feel free to add me on LinkedIn if you want to connect, but that's what I had.
Thanks, Miko. Wow. Andrew, you had completed a degree too before you started community college, am I right?
Yes. So my degree was actually in computer science. So I was kind of totally changing fields when I got my associates in biotechnology. But yeah, I'm very glad I did that. And I felt I had way more opportunities for extracurriculars at the community college and for biotechnology than I ever had for computer science. So yeah, I think it worked out.
That's great. I have one more quick question too before I open it up and take questions from everybody else. And that is, so you used IC&3D in the hackathon, and then on your job you were using Pymol. Did you find that your experience using IC&3D helped you learn a little bit about Pymol or understand what was going on?
Yeah, I think the biggest thing was already having that knowledge of knowing what I'm looking at, basically. So I had already experienced looking at proteins and trying to identify specific things. So really the only difference was the interface. Pymol has different interfaces and controls, but other than that, yeah, it was essentially the same process that I did in IC&3D. So that exposure definitely helped.
Thanks. I mean, the thing we always worried about when we're teaching is, do we need to teach the exact thing that somebody's going to use, or can they transfer what they've learned and apply that to a new situation?
Yeah. Yeah, no, it was very transferable.
Great. Uwe.
Thanks. Thank you for the presentation, guys. Listen, a couple of questions in the same realm, having to do with how did you become aware of what types of internships you wanted to pursue? Did you have an advisor? Did you find interesting publications? Was it just what was available? And that leads to the second question then is, how did you identify openings that you could pursue? So how did you realize in what areas you wanted to pursue internships, and both of you, and then secondly, how did you become aware of openings? Thank you.
I can start.
Sure.
So for me, the exposure at the first hackathon is what led me to pursue more opportunities in antibody engineering. And most of the opportunities I found out about were through my professor, Dr. Bryans, who I owe a thank you to. And also the internship I did was at the Wistar Institute, and one of the deans of undergraduate studies was on my team at the hackathon. So that was a really good connection for getting into that internship and being exposed to that. And then I did research on like what companies they're partnered with and what labs they have available at the Wistar Institute that focus on protein and antibodies engineering. And so I was able to intern at places that gave me that experience.
That's great. Miko.
Yeah, I think I'm going to say that it's really good to be able to like network and join like these kinds of events. Like in my very first internship, how I got that was I participated in a number of like networking events, like speed networking events that that CCSF offered, and I joined their internship program, where you get into their internship program and then you can start talking to other labs to kind of like grow interest, because they have partnered with a number of labs in like the Bay Area, and they're interested in taking interns maybe like 2 times a year. So I was able to like talk to a number of different labs there. They're all studying different things. That first experience was in antibody engineering, but there are other labs like studying things like bioengineered meat, for example. But I think for me, the way I did it was I did have like an interest in like healthcare, for example. So like that narrowed down quite a bit. And then from there, I was pretty interested in immunology, so that narrowed it down even further. And to get to like, I guess, antibodies and wanting to learn how to use them, like learning about the immunology piece really got me to make that next jump. I'm sorry, I hope I answered that question. Great.
More or less. Other questions from our audience? Okay, well, I have a question. Well, people are thinking of questions. So many of the scientists I know, and myself included, would almost rather drink raw sewage than network. And so I'm wondering, what did you do to learn how to network, or how to force yourself to do that? And I'm going to start with you, Miko, and then Andrew.
Yeah, I think that part of it was like actually being like on the floor, like in the lab. Like sometimes you don't have all the guidance in the world there, and you're just kind of there, and you're just going to be there. And maybe you need a little help because you're lost. So it all started like in lab. So like maybe I'll ask someone for help, and just kind of like start building that connection. And maybe that person doesn't know what to do. Maybe they can point you in another direction. And it just kind of started from there, and really getting involved with all of the people within like my lab, maybe my institute, and also like my program there. Like you can take like these small steps. Maybe you don't even really have to bother them about a lab problem. Just as small as just like starting like, what'd you do this like last weekend, or something like that. But yeah.
Andrew, how did you force yourself to network?
Yeah, it's good that you said it like that, because I definitely had to force myself. I fall under that introverted scientist camp. But I think probably the biggest benefit for me was actually in my undergrad for computer science. There was a heavy focus on networking there. They encourage you to like build a nice LinkedIn page, make your own website, reach out to anyone and everyone at any company you might ever consider working at. So I think just through that repetition, it eventually made me more comfortable with networking with people. And I think one of my like go-tos is usually connecting with professors. They usually know a lot. So I think they're very valuable resources, and that proved true for me.
Thanks. Well, speaking of professors that know a lot, I'm going to start with Dr. Bryans.
Hi, Andrew. This question is really for both Andrew and Miko. So you both sort of learned new software, protein analysis software during the last hackathon. Can you comment on that experience? You were kind of learning how to use the software and actually using it to provide us with data in a very compact, maybe under pressure situation. So can you comment on that, both of you, if you like, comment on that experience of the last hackathon and what you learned?
Yeah, so for me, luckily I have experience with computers, and also this might sound strange, but I think playing video games actually gave me an advantage in like picturing that protein in like that 3D structure in my mind, and like knowing how to manipulate it on the screen and what I'm looking for. And yeah, I think the like pressure, the like time constraint of only a couple days that we had to work on it, that really forced me to sit down and try to learn all the quickest ways to get the information I needed. And I wouldn't say it made me a pro with the program, but I'm familiar with it now to where I can pretty much get what I need rather quickly. So I think the frame of the hackathon, kind of forcing you to focus on one thing for a couple of days, really helped in learning that software. And then my previous experience also helped me with that.
Yeah, you quickly became the expert on the team, I remember.
Yes, yeah.
So well done. Miko, did you have a similar experience where you had to learn it in a short period of time and actually put it to use?
Yeah, my experience at the hackathon was my first time using that software as well. I agree, like having that really condensed amount of time really put a little bit of pressure on our group to learn. But I think like the aspect of having it being used as a teaching tool, like made it an easier approach to like actually learn how to use. Because I guess when you try to figure out how to explain how to use a tool to someone else, like you make all those connections yourself as well, which helped out.
Thank you.
I feel bad in some ways, Andrew, because there are also a lot of ways to access data from ICN3D programmatically and through scripts like Node.js that we never told you about.
That's all right. I'm fine not doing extra coding.
But Jayao has all these things on the GitHub page. So if you ever want to go back and look for that, it's all there.
Yeah.
Daniel.
I think it was Miko that in his advice list, he said good lab habits is something that he would say you should develop. So I'd like to hear from both of you if you could tell us more. What do you think of as good lab habits? And did you have a classroom experience that really led you in that direction that we could try to replicate for other students?
In terms of, I guess, it could be as small as just like having good aseptic technique. I know that's something that programs always kind of enforce a lot, but good habits in the sense also of maybe understanding like the lab culture there and not having to really like work yourself all the time. And I think it'd be good to kind of convey to students like what a lab culture can be, because maybe you just think lab is all about work. And maybe you're there like 6 days. People think you're there 6 days a week. You're just trying to churn out data all the time, but like really have like trying to have like good work habits, like be there like you're working a job and not just, or a job and like have a life at the same time. And it's not really always about the work, but it's about having like these good habits when approaching work.
So yeah, for me, I think the best lab habit you can develop is being a good what's the word I'm looking for? I don't know, just being a contributing member of the lab. Like I think everybody, no matter what level they're at, is responsible for restocking pipettes, pipette tips, making sure there's like, if you use the last of a reagent, making sure that gets remade. I think that responsibility really falls on anyone in the lab, which might not be something that students are aware of. They might think, oh, like high up doctors don't do that work. But I think from my experience, I've seen that everybody really does all that work. And I think an experience in the biotech program that Dr. Bryans teaches that was beneficial. We did a pipette challenge and pipette verifications. So the pipette challenge, you had to get your pipette volumes basically within, I think, 2% of what you were trying to get. And then also like we did like cleaning and verification of those pipettes. So like understanding how they work and, understanding like why accuracy and precision are important. I think those activities that we did really helped build those lab habits of always focusing on your pipetting. And yeah, I think those are really beneficial.
Okay, I'm going to have Todd go next, but I see we have several students here. So students, this is your opportunity to, ask questions of people who are just a little bit farther than you down the road. So I want you to be thinking of things. Okay, Todd.
There we go. Yeah, how about notebooks as part of lab habits? Can you comment on that? Also comment on what are kind of the current industry practices in notebooks and management?
Yeah, if you have the opportunity to learn how to use an electronic lab notebook, that is something that would be a really good skill. But if you guys aren't doing that, like even just learning how to keep a regular, like at work right now, I use a regular, I use a online lab notebook, but I always keep like a small notebook with me also, just to jot notes down and just have with me at all times if anything happens. But yeah, learn how to use electronic lab notebooks. I can, if you have more specific questions about them, I could go into it too, but I'll let Andrew go.
Yeah, so big focus of Dr. Bryan's program is handwriting lab notebooks that are up to par with industry standards. So there's a handwritten lab report that you need to do for every class, every experiment. So that gave me a lot of exposure to writing lab notebooks. And then at the company I'm going to work at, while I interned there, nobody had that like lab notebook that I was writing up, but there are like SOPs that get printed out that have information that you need to fill in as you're doing your experiments. Every like barcode, every reagent and pipette is like barcoded. So the lab management team does like inventory of that every day by checking those barcodes. And yeah, I think there wasn't as much like handwriting of notebooks in that lab. Really more just filling out SOPs and then like managing inventory through those barcodes.
Yeah, I will say it's like being able to learn how to make those handmade lab books in the beginning like really slows you down quite a bit, and you really understand what's going into your notebooks.
Mhm.
So that's also really something valuable you could have.
Yes, if I followed up, kind of the practice of doing a handwritten notebook transfers well to using electronic and other formats, and kind of in establishing the importance of record keeping. Is that fair?
Yeah, definitely. And it exposed me to like the kind of information that I need to be looking out for, that I want to document for any experiments I'm doing.
And
Cool,
just
thanks.
to follow up on that a little bit more, have either of you had to write an SOP in your positions?
I haven't written an SOP at my current job, but I was a lab technician at a company that made supplements for a little bit, and I wrote some SOPs there.
I haven't been here long enough to have written an SOP, but I will have the opportunities in the future, but and also to like audit other people's SOPs just to like try them out, see if there's like anything that it can improve upon before it actually becomes an SOP within like our lab group. But yeah.
More questions? Oh, Peter.
Andrew and Miko, thank you so much for sharing your stories. I've got a question about choosing community college technician programs. Both of you had bachelor's degrees. What did you get out of those community college programs that you didn't get in your bachelor's degree? Andrew, if you'd like to go first.
Sure. Yeah, so I got way more out of my community college experience than I got out of my bachelor's degree. The opportunities available were much more plentiful. Dr. Bryans is really well connected in the area, so she was able to connect students with internships. I think 7 or 8 students all participated in the Wistar internship program that I also just completed. And I think also like the professional certificate program, or like part of what you receive when you finish the biotech program at the community college, is really beneficial. It was like professional training, so it really prepared you for a job in industry, as compared to liberal arts computer science degree. I learned coding, but they didn't really prepare you for any specific field, and they didn't really teach me what opportunities were available. I felt that at the community college, Dr. Bryans really presented like all these different areas of the biotech industry that you can work in. If you don't want to do lab work, maybe you can do QA, QC. You know, just a lot of opportunities.
Yeah, and I'm gonna go off of that. Even beyond like opportunities, I think that at the community college level, like these programs are a lot closer to like maybe a lot of these like biotechnologies, and like they work closer with the industry, so you can get a lot of know-how and like insight into what these companies maybe are looking for in terms of skills. And there's a really big focus on like all of these skills and learning how to apply these skills to whatever job you might want to go into.
Okay, other questions? Students, come on. So many things. All right, well, I have another question. One thing that we wonder about, we know from our own experience, well, a lot of my students, when they interviewed for positions, they were asked to do things like, I don't know, describe how you would make a 2-molar solution of NaCl, or pipette, or there would be some sort of practical thing they had to demonstrate that they knew how to do. Did either of you have things like that in your interviews? Somewhere where you had to demonstrate that you know a particular lab skill or piece of knowledge?
So I think since I was interviewing for a full-time job while I was interning at the company, the interview process was a little different. So they didn't ask me specifically, like, do you know how to use a pipette? Because obviously they were teaching me that currently. But they did ask me, like, if I knew some of the procedural steps for some of the experiments that I was doing during the internship. So there definitely was an aspect of practical lab work that I needed to demonstrate in the interview, but nothing like as broad as, do you know how to make a 2-molar NaCl solution? I think because I was already interning there, so my experience might have been a little different than someone who's applying to a company that they don't have any experience with. But yeah.
What about you, Miko?
Yeah, I guess my advice on something like that would be really understand the question before you try to just start blabbering something out. Like you can clarify like any question, but on the more technical side, the current position I'm in does use like flow cytometry a lot, for example. So I did have to have like some basis in flow cytometry before I could actually apply for this particular job. But on top of that, like more regular like lab, more everyday kind of stuff. I don't know if I was necessarily asked about something like that, but my advice, yeah, would be to really understand the question before just kind of going at it. And they're okay with that usually during interviews, I hope.
Oh no, I think that's really good advice. Dua.
So, as a researcher, are there any challenges or opportunities that came up over time? Because in the start, I mean, I've done one internship so far, and there were just basic struggles that I had that I overcame in my own way. But over time, what are the new like struggles or opportunities that are down the road, would you say?
I think for me, so since I've started like this new job, I had been like bouncing around between like internship, like all these like small projects, and it was really cool like being able to get involved and be like, oh, that's a really cool project. I'm gonna like do my little part in it. But now that I'm here like for a full-time job, the length of which I'm gonna be here is gonna be a long time, so I can't like be super excited about like this project. I mean, I should be excited about this project the whole time, but like being able to like celebrate like the small milestones in like your projects, for example, like being able to understand like why you're there, and you're not just there for, I don't know, like 6 months, 2 months, and you're just going to make it to the next job. But yeah, I guess I hope that helps.
Yeah, I think I haven't had the opportunity to do any like independent research yet, but I think from what I've experienced, the biggest hurdle I've had to overcome is accepting failure, because there's a lot of failure in researching. Even at the company I work at, all the processes are optimized to like work every time, and you'll still have projects that don't work with that process. So I think accepting failure, troubleshooting, and understanding why something failed is definitely something that's very important. And then understanding what your next steps need to be.
Gotcha. That makes sense. So like bouncing back up. That's cool.
Yeah.
Todd.
Yeah, I like the things don't always work, and it kind of ties into what Miko said earlier was I've learned you don't have to know everything. So can you comment a little bit on what you've been learning in terms of how to troubleshoot? And I like to say I don't fail anymore. I just learn. There's a kind of just a way to approach that.
Yeah.
Can you comment on just some of the things that you've developed that you weren't taught these things, but they're going into your experience now and maybe forward?
Yeah, so I guess for me it would be if at my internship, if I ran an alanine scan on a certain epitope, and then after my flow cytometry I got really weird results. Maybe I didn't see any dropouts to identify residues where the antibody might be binding. I think like understanding what steps to do next. So like knowing to either increase the pH, decrease the pH, stuff like that.
I think for me, always ask questions, especially like if you're early on in your process or a new job. I think my boss likes it right now when I'm like asking questions, making sure I know what I'm doing before they let me out and just kind of operate on my own. Ask as many questions as you can, like in the first however long your training is. But I think being also being able to leverage the experience other people have on your team is super important for learning, and you can develop your understanding for your project or how your team operates so much faster if you're able to talk to these other people and figure out what's going on.
Thanks. I think we have time for one more question. Do we have another? Peter.
I was just wondering, Miko and Andrew, what did it feel like the first time you walked into a biotechnology lab? Do you remember what it felt like when you walked into a lab the first time?
Honestly, I felt like I was in a movie or something. I mean, the lab had a lot of robots, and I was like, whoa, this is really futuristic. I've never experienced something like this. I mean, we had good resources at the community college, but walking into a company that has really top of the line resources, it was a little overwhelming, but also really cool and rewarding to feel all the work I had done had finally paid off, and I get to work in this really cool high-tech lab.
For me, my first experience into a research lab wasn't in industry. It was it was in academia. So when I walked into there, I actually wasn't, it looked kind of the same as like some other labs I've been in, like on the academic side. Maybe the equipment was a little different, but like there were still benches. But what the cool thing for me was to see other people, like they were like graduate students and stuff, but there were like people like my age-ish there, and like they were real people, and like it seemed like something that was like feasible for me to like be in. They weren't all like really like well-seasoned scientists there, and I would just be like the new guy or like the new like kid there. But yeah, that was kind of cool to see.
Thanks. Well, I have to say, we could probably keep you here all day just grilling you about your experiences and learning, what obstacles you've overcome and all the wonderful things you're doing. But unfortunately, it's the top of the hour here. So now I thank you so much for coming and talking to us and sharing what you've been doing. This is so great. And everybody, thanks.
Oh, yeah. Thank you for having this. Great opportunity to learn.
Oh, I think we all learn, even those of us who've been doing this 30 years are learning a lot. So now what we're going to be doing is I'm going to turn the recording off.